Hello. Hello.
Hello.
Am I audible to everyone?
Yes, ma'am.
Yes, ma'am.
Okay. Let's start. Just a second. My slide is visible for to everyone.
No,
Yes,
ma'am.
ma'am.
Just a second.
Yes, ma'am. It's visible.
Visible. Okay.
Yes, ma'am. It's visible.
Just a second. It is problem or not? Hello.
Hello.
My
Hello,
slides are
ma'am.
visible.
Yes, ma'am. You are audible, ma'am.
Okay. So,
Ma'am, your slide is not visual.
not visible. Just a second. It is already showing that it is visible to everyone. Let me reconnect it again. Now, is it visible to everyone?
Yes, ma'am, visual.
Yes,
Okay.
ma'am. Visible, ma'am.
So, first of all, I would like to welcome you all to attend this webinar. Here's myself, Dr. Shweta Tripathi. Actually, this webinar is related to NGS data analysis, which is very useful for any biotechnologist or any biotechnology background or non-biotechnology background people to create their new skills and enhance their future prospects or their skill development. Second thing, in this program, we are starting from theory to the hands-on session. During that, we will study about the sequencing technologies, which are the sequencing technologies involved in NGS. which was the databases, tools, softwares, programmings are involved in NGS data analysis. So, this can be useful to create your own criteria related to NGS data analysis. During this program, we are going to start from scratch to advanced level, which is very good to improve your skills. During this program, 15 days, during this program, if someone are saying slides are visible, some are saying not. What is the issue in that? Not visible for everyone slide right now.
Ma'am, we can see your web browser, but not the presentation, ma'am.
Okay, just a second. Now, is it visible?
Yes, ma'am.
Yes,
Now.
Yes, ma'am, it's visible,
yes,
ma'am.
ma'am. Now it's visible, ma'am.
Okay, just a second. So now is it visible to everyone right now?
Yes, ma'am.
Okay. So first of all, I would like to welcome you all to attend this webinar. This webinar is totally related to NGS data analysis. Here we are going to clear some doubts, some concerns, some points, some issue related to NGS data analysis. So we are going to take 15 days training program related to NGS data analysis. During this course, we will provide you the guidance, the theoretical knowledge, and hands-on session related to NGS sequencing, how it works, NGS databases, their various tools, softwares, how to design a particular workflow as per your interest. So here we will provide you some knowledge, and during this 15 days program, if you have any doubts or query or any concern, you can just mail me or message me. Will help you out to solve that problem. During this program, you can means if you are a technical person related to bioinformatics or non-technical person, but easily you can understand this NGS data analysis. I will try my best for this. This is all about 15 days training program. Here I will now brief about myself. Myself is Dr. Shweta Tripathi. I have done my BSc, MSc, MPhil, PhD. So my research area is cancer informatics. After my academics, I have started research work in different academics, labs, industries. uh, During that, we worked on Alzheimer, cancer, mushrooms, etc. So we worked on R language, meta-analysis, NGS data analysis, and various genomics and proteomics related research work. So here I am very happy to share my knowledge with you all. It will great help to you all to understand the basic of bioinformatics with NGS data analysis. So again, it's immense pleasure to welcome you all to attend this webinar. Am I audible to everyone?
Yes,
Yes,
ma'am.
ma'am.
Yes, ma'am.
Yes,
Oh,
ma'am.
okay. If any problem, just unmute your mic and you can just speak it out. Okay. So after this 15 days program, what we are doing is during this 15 days program, we will actually study very broad prospect of NGS data analysis. NGS data analysis is not specifically to a particular field. Actually, it is the broad class. In this, you can carry out further research in the area of cancer, transcriptome, metagenomics, whole exome, and RNA sequencing. So now come to the topic: next generation sequencing and data analysis, unlocking the power of genomics. As the name suggests already, next generation sequencing means sequencing. You already heard this term. before. That was DNA sequencing, but this one is the next generation, means the more developed, more advanced as per recent trend. And data analysis after that sequencing, there are lots of data produced. So what we can do, we can just analyze them and we'll come to the conclusion for a particular result which is helpful for various scientific community. So this is all about the NGS data analysis which we need to perform, we need to learn. So here is the some briefing about next generation sequencing. So actually, initially the sequencing is carried out on small platform, time consuming, lots of time, lots of money, lots of reagent, etc. But right now we can run multiple experiments at a time. So what we can do, actually this NGS is highly massively parallel, high throughput massively parallel sequencing which runs thousands or millions of reads at a time. It is just like center sequencing follow the capillary electrophoresis. So here is the briefing. First, you have to choose the genomic strand. Then we proceed for fragmentation, ligation of fragments, and base of each fragment is identified with the emitted signals. Actually, this NGS removes the gap between the micro and nanotechnologies. It is reduce the sample size, low cost reagent, less time, and very effective result. You can easily analyze any data, millions or thousands of data at a time, and very useful and important for scientific community to come to the conclusion and helpful for the human being. So here is the pictorial diagram how NGS works. So first clinical sample. So this clinical sample or any sample of interest, whichever you want to take, just carry it out. After the sample, you have to extract DNA or RNA of your interest, DNA or RNA of your interest, the further you can carry out the NGS experiment. But this NGS is broad, so you have to choose which one you want: WGS, whole genome sequencing, RNA sequencing, or transcriptome sequencing. Among three, whichever you have interest, you have to just choose. After this, they produce the sequencing data, and this data is further proceed for the data analysis, where bioinformatics used to work actually. So this comes to the part of bioinformatics. Here you can see after sequencing data analysis, you will get genome assembly, gene expression profile, and phylogenetic classification. And based on these results, you can conclude or you can come to the conclusion also that this can be useful for the clinical diagnostic, biomarker discovery, outbreak control, resistance testing. See, in short term, I can say NGS data analysis is not a simple task. You are performing, you are analyzing data, but that doesn't mean it's a particular useful or particular fruitful result. Actually, if you are going through a particular work plan, so it makes your task easy to justify that why and what you are doing right now, is it useful to the scientific community or not. So this is the basic program, basic understanding of this 15 days program, that if you are performing any NGS data analysis, so how it is useful for you to link in your research or link in any industrial purpose or link for your area of interest. So you can come to the proper fruitful conclusion which can be useful for the research purpose. Next slide. So there are lots of technologies and developmental changes occur in NGS technologies based on their time, cost, template preparation, sequencing, visualization, and data analysis. So here I mentioned top six technologies. First one is Illumina or Solexa. Second one is Rose 454. Third is Helicos Biosciences. Fourth one is the Light APG Solid System. Fifth is Specific Biosciences. And sixth one is the Ion Torrent technology. See, these top six technologies pictures are listed here. You can just visit any research center, any industry. So you can check this kind of machineries are available. So you can just visit over there. You can just come to know these systems are working in the field of NGS. So here is the brief comparison of all the NGS technologies based on their different criteria. So first, if NGS technology starts from the material. So here you can see the rules 454. took the 20 mu g total RNA or DNA, with same Illumina and ABI solid. So here is the how much sample you can take for the run. Amplification generally Rose 454 and ABI solid use the emulsion PCR based method, while Illumina used the bridge PCR method. The third criteria is support of template. So you can see Rose 454 use the beats, AVI Solid uses the beats, but Illumina uses the flow cell surface. Fourth criteria for this technologies are the enzyme use. So you can see the sulfurylase and luciferase used for the Rose 454, while DNA polymerase used for the cluster amplification for Illumina technology, while DNA ligase to ligate the prob hybridized product. Fourth criteria for these technologies are the sequencing chemistry. So generally, the Rose 454 works on based on the pyrosequencing, while Illumina works on by the sequencing by synthesis, and ABI Solid works based on the sequencing by ligation. So surface area of sequencing reaction, generally Rose 454 use the microtiter plate, while Illumina uses the channel flow cell. ABI Solid uses the glass slide support detection. So after fragmentation, they used to dye those fragments. So here they are using the fluorescent dye, reversible fluorescent dye terminators. They are using while Illumina and ABI Solid uses the chemiluminescence detection. Read length varies as per the platform. Rose 454 uses the 400 to 500 bases, while Illumina uses the 35 to 40, 50 base, and ABI Solid uses up to 50 base. Positive aspects of this technologies are Illumina is the low cost and less material, starting material, and are needed than the four, means it is compared to Rose 454, Illumina is more useful compared to ABI solid also. So negative aspects of Rose 454 is higher cost per base, while in Illumina you will get the shorter read, but the cost is low. While in ABI solid, the shortest read and very new instrument. So we cannot take risk on that. Illumina is very trendy, it is very useful. Why I listed this comparison on the slides? Actually, the reason is that if you proceed for NGS data analysis program, so you have some basic understanding and basic comparison of this technology. So anyone can easily, I ask you why you are doing, why you have chosen this platform for sequencing and all. So this is the basic thing you have to understand. So I listed here. So next slide. So now come to the main point, which is part of our biohealth learning program, NGS data analysis. So here is the broad classes how we are performing the NGS data analysis. See, every technologies are available, but after these NGS technologies, they used to proceed for the sequencing. If they proceed for sequencing, definitely they will produce the results. So these results known as the raw reads. They can be in FASTQ format and other formats, which I will explain in further slides too. Then further we proceed for quality control, then quality trimming, and the fifth one is the alignment or gene mapping. So this is the broad work plan for NGS data analysis. This can be further classified as per your interest also, which I will explain in further slides also. Now, so your doubts and queries must be clear. So we learned about different technologies, sequencing and platform. So after sequencing, they used to produce vast amount of data. So we need to submit store it in a particular places. So here are the list of repositories for NGS datasets where we can submit it freely. So first one is the master one, that is known as NCBI, National Center for Biotechnology Information Database. Inside this, there are the three subclasses of database. First one is the sequence read achieve (SRA). It is actually a public repository for raw NGS data. Then gene bank. Gene bank generally can contain both DNA and RNA, both the NGS sequences data you can get from here. So third one is the bio project. It is also part of NCBI, which contain the information of biological information, including the NGS. Means you have NGS data with their particular biological information and description also. Second repository is the European Nucleotide Archive, ENA. So it is the comprehensive database, storage and retrieval of biological sequences. Here you can also submit your NGS data or also retrieve from here. Third one is the DNA Data Bank of Japan, DDBJ, a biological database that collects and provides public access to DNA sequence data, including NGS data also. Fourth one is the Array Express. See, in next slide I will explain Geo dataset browser also. So Array Express and Geo datasets interlink with each other. Means you can search and you can directly go through the Geo dataset browser also. A database generally Array Express contain the functional genomic experiments that includes the NGS data, microarray data, and other high throughput sequencing data also. As functional genomics means, suppose any cancerous cell line is there, they are treated with particular natural compound or any drug active compound, and they submitted that experimental data or NGS or DNA chip or NGS data format, file format. So you can just retrieve from there also. If you want functional NGS data sets, you can also get it from Array Express. It is freely available here. Fifth database is the Gene Expression Omniverse. Actually, it is also known as the GEO dataset browser. You can also get functional genomics data from GEO. Sixth one is the Cancer Genomics Hub, CG Hub. So here, as the name describes, cancer genomics means you can get all the data related to cancer. Means all type of cancer data you can get from here. You can also get some tools and softwares here, so you can analyze direct those data using various softwares and tools related to cancer. The seventh one is the European Genome Phenome Archive, EGA, which contain the human genetic and phenotypic data. Means it contain both the information. So you can, based on that criteria, you can also retrieve the NGS data. The eighth one is the International Nucleotide Sequence Database Collaboration, INSDC. So actually, it is the partnership between the DDBJ, Gene Bank, and ENA, which ensures that the comprehensive storage of DNA sequence data, including the NGS data. So these are the most important eight repositories where you can retrieve your all the NGS data or microarray data or any FASTA file, sequence file based on your interest. What you have to do, just go on the web page, homepage of the database, click on that, search the term whichever you want, and you will get the list of it and filter it out and retrieve the NGS or any interested data related to genomics from there. Next slide. So now, after sequencing, those data were submitted into the various repositories. So now next is the means if you are getting this, so this data set must be in particular file formats. So here are list of formats which is related to NGS only. So first one is the FASTQ, the most common file format of NGS data sets. So this is the most common format for storing the NGS data, which contain DNA or RNA sequence reads along with their associated quality scores. Well, second format is the FASTA format. It contains same thing, DNA or RNA sequences, but without quality score. See, this is difference between the FASTQ and FASTA. FASTQ contain the quality scores, while FASTA does not contain any quality scores. It includes the nucleotide sequence information in a plain text format. So these are the two most common format of NGS dataset. Third one is the SAM or BAM. SAM stands for Sequence Alignment Map, and its binary counterpart, and BAM stands for the Binary Alignment Map, and these are the formats storing the sequence alignment data. See, SAM is the human readable format, while BAM is the machine related, means as the name suggests, binary version of the same data. So these formats contain the align sequence reads and associated the metadata. So this is the new term you come to know that here you have lots of data, but what is metadata? Means you have multiple NGS data, but among those data, you are getting more common data among. Suppose you have 10 set of NGS data, 10 NGS data sets. In this data sets, you are getting 10 most common files. So those data are known as metadata. So this term is called metadata. Next. So fourth file format of NGS data sets is VCF, variant call format. Is used for storing the genomic variations such as the SNP, single nucleotide polymorphism, and the small insertion or deletion. Includes information about the genomic positions, reference sequence, alternate alleles, and a variety of quality score. See, when you proceed for sequencing, so you have to choose which file format you want. At the end, you will get those file formats. So based on your interest, you can choose and you will get the result in that file format only. Fifth one is the BAT file format of NGS data sets. BAT. BAT stands for Extensible Data format is often used to represent genomic intervals or regions of interest, and it is generally for chromosome coordinates, start and end position, and optimal additional information. So every file format have their unique information, which information you want to retrieve and analyze. Sixth file format is the GTF or GFF. GTF stands for gene transfer format, and GFF stands for the general feature format. So generally, it is useful for the genomic annotations, which contain information about genes, transcripts, exons, and other genomic features. Seven file format is the CRAM, compressed SAM. Is a binary format for storing aligned sequence data. It offers better compression compared to BAM while maintaining the ability to retrieve original read information. And eighth one is the last file format of NGS dataset, that is Big Wig or Big Bang. So actually it is used for binary format used for the genome wide signal tracks and read the coverage or chip sequence data and is allow the they allow sorry, they allow efficient storage and retrieval of large scale genomic data. See, if you visit any database, you just search and based on your interest, suppose for example, you want particular SNP data. So you just go on that homepage of database, search on that, and just choose the VCF file format, which will provide you the information of SNP, and you can further use that and analyze that data. So those criteria you have to fix which file format, which data you want. So these this is all about the repositories and file formats of NGS datasets. Now sequencing done, sequences retrieved, sequences stored, their file format understood. The next step is the data retrieval steps. The first one is the identify the data source. So this is actually in short term. In briefly, I can say this is the brief overview how you can plan your interest or research for NGS data analysis. So first, identify the data source. Means suppose you want to perform any research in the field of Alzheimer, or suppose recent trend is the COVID. So for this, you need to choose which data you want. So first, choose a database. Among those database, you have to choose suppose NCBI, SRA, SRAGEO, etc. Second, access the database. After selection, you have to just go through that website. Some websites are there which can help you to retrieve data directly, while others put some condition. You have to create your own account and retrieve the data. So access the database. Second is important criteria. Third one is the search for the desired data. So if you are proceeding for further, means you have selected the database, you access the database. So now you want desired data. So what you have to do, you have to just search a particular term, any project ID, any sample name, any organism name, or any specific keyword which you want as per your desired result. So you can just type over there and click on search. After this, means you have search. So fourth thing is the evaluate the search results. You will get multiple results. So what is problem in that? You have to choose the specific one. Which one you have interest in, which one you want. So in this, you have to review the search results to ensure that the identified data matches your requirements. Pay attention to the metadata such as sample information, experimental condition, data quality matrices if available. So these are the things to choose a specific data, choose a specific database, choose a specific experiment, choose a specific data you want to retrieve. So these are the four main basic steps for NGS data analysis you have to start. Next is to select the data files. So this data files you have to retrieve. Initially, generally they will provide the FASTQ file format, general format. Second is the BAM file. But in any case, suppose you want to analyze the data and that software or tools is not accepting this format. So there is another option we have. You have to convert that format to the acceptable one. Suppose that software wants faster file, so you can convert that FASTQ into the faster file format and then further proceed for the analysis. Sixth one is the download the data. So follow the instruction provided by the database to download the selected data files. And typically there will be option to download individual files or complete the data sets. And consider the size of the data and available storage space on your computer or server. Seven is the data format conversion, which I already explained in fifth step. Eighth one is the data pre-processing. See, if we are performing we retrieve the data, so during the sequencing procedure there are lots of error, there are lots of impurities. So for this we need to proceed pre-processing. In this what we are doing, we perform the quality control, trimming adapters, filtering low quality reads, or performing other data cleaning steps, so that your data is pure and you will get accurate results. So it is most important step to proceed for the data NGS data analysis. Ninth one is the analyze the data. So you can proceed for the downstream analysis of NGS data like genome assembly, variant calling, differential gene expression analysis, or specific analysis based on your research goals. So this is all about. So first, I hope you have a clear concept about how you can analyze NGS data. Yeah, there is a in previous slide I already explained the brief note on that, their technologies, their repositories, their formats, and the brief work plan for NGS data analysis. So applications of NGS technology. The first one is the human genomics and precision medicine. This is gene discovery, where we can do the whole genome and whole exome sequencing used to identify genetic variants. Next one is the pharmacogenomics. So, as the name already suggests, pharmacogenomics. Pharma means drug and genomics means related to any genes. So it is actually suppose any cell is treated with a drug and you want to perform NGS, you perform the NGS analysis. So you can what we can get the result. You can actually study the drug response, metabolism, the toxicity. Based on that, you can just improve the outcomes. Like you minimize the adverse drug reactions, enhances the patient safety. Third application of NGS technology is the cancer genomics. So NGS has transformed our understanding of cancer by enabling comprehensive genomic profiling of tumor. NGS-based tumor sequencing allows the identification of somatic variation, mutations, copy number alteration, gene fusions, and driver mutations. Next application is a non-invasive prenatal testing. So here, what we can do, we can just understood the fatal genetic abnormalities. Initially, which kind of chromosomal problem can be occur in a child. For example, Down syndrome, Adverse syndrome, and Patau syndrome. So this is also useful application of NGS. Next one is the rare disease diagnosis. So NGS is already means very useful in the field of health and diseases. So here what we can do, the whole genome sequencing and whole exome sequencing that uncover the disease causing mutation, and it is very useful to understood that rare disease uh, clinical features. Next application is the population genomics. In population genomics, suppose you means multiple. Suppose best example is the COVID. So actually, this COVID symptom is differ person to person. Some have a coughing issue, some have suffocation issue. So based on the comparison, you can take sample of individual and run at a time, and you will come to the conclusion of what are the gene which is responsible of that. So it can be useful to understand the human genetic variation and population genetics. So these are the actually very important use of NGS data analysis in the field of health sector. Next is the cancer genomics and personalized oncology. So first is to create the tumor profiling. This tumor profiling, or in short term we can say DNA chip. So it is useful for the further understand the further progression of tumor, and you can treat a person using gene. Next one is a targeted therapy selection. So targeted means critical role in identifying a specific genetic alteration in tumors that can be targeted with existing or emerging therapies based on the driver mutation or biomarkers. So if you identify a particular gene which is responsible to cause a tumor, that can be a biomarker or driver mutation. So next application is the resistance mechanisms and treatment monitoring. So, NGS allows the monitoring of the genetic changes that contribute to treatment resistance and disease progression, and serial monitoring of tumor DNA through liquid biopsies for tumor detection. So this is another application of NGS. MRD, the minimal residual disease, means those diseases which is not easy to capture. It is very microscopic. It is very rare to catch. So for this, NGS is also useful. Next is the immune profiling and immunotherapy response. So you can enhance the effect of, you can just boost your immunity based on the tumor microenvironment and assessment of immune cell infiltration and checkpoint of molecule expression. Next important application of NGS is the germline variant analysis. So it is actually identify the germline variants associated with cancer predisposition syndromes. Means it is very advanced. You can just identify at the bottom of the disease cause. Next important application is we are performing the NGS sequencing part that is known as the metagenomic sequencing. What they are doing, they perform any means isolated species sequencing, and based on that, they understood they are understanding their biodiversity, their species interactions, and ecological processes. The fifth important application of NGS data analysis is forensic and paternity testing, where what we are doing in forensic, we can just collect the sample from the crime scene, so you can easily determine the relationship between the individual and resolution of complex sensitive cases. Paternity, suppose someone is like blaming this child is mine, this child is mine. So in this case, and this is also useful to identify parenting, whom identify the genetic and identify the paternity testing. So next is the career opportunities in NGS sequencing. So first one, most important and the basic level of starting your career in the field of bioinformatics is the NGS technician, just like a laboratory researcher. So what they are doing initially, they are going through the laboratory preparation, like they are preparing buffers, solution for NGS experiments. Then they are checking the operations of various platforms like Illumina, Iron Torrent, His Bio, Oxford Nanopore system. And after that, they set the overall instrument, run initiation, proceed for the sequencing. Third important role they are doing to prepare the sample means they are isolating the RNA or DNA, extracting library construction, quality control, and they follow the standard protocol to maintain the accurate documentation of sample processing steps. Fourth one is the quality control and assurance. Responsible for conducting quality control checks at various stages means the overall workflow is right or wrong. So they use that work is done by NGS technician only. So they can also maintain the troubleshooting and data management. So this is the task performed by NGS technician at any research center in the industry or at a particular lab also. Next one is the means, bioinformatics scientists or research scientists. What they are doing, actually, they are the master one. They are designing experiments based on their requirement, based on the funding. After that, sequencing the data is produced in various file formats. So what they want in their results, based on that, what they are doing, data sequencing data, performing data mining, identifying genetic variants, and interpret the results in context of research question or clinical application. They collaborate also with the bioinformaticians for more complex analysis. Means basic one they can analyze, but if it is more complex, they use to take help of any software engineer also a bioinformatician. They also work in the field of R&D, research and development, to develop new protocols, methodologies, and workflows. Uh, Evaluate and implement the emerging NGS technology means the further development is carried out by them only. Means daily new and new changes are carried out. Project management. Then there are lots of project ongoing in the under him or her, so they can just collaborate with multiple places to finish on time. So these are the tasks done by the any research scientist of bioinformatics. Next important career for the bioinformatics is the bioinformatician and data analysis. See, in previous there is the technician. Now they are CN, bioinformatician, means they are just analyzing data, developing softwares, database, etc. So what they are doing? First one, they are doing data analysis. So in this analysis, what they are performing? Quality control, alignment, variant, calling, and annotation. And also they are using various softwares and programming language to extract a meaningful information from the raw data. Second, they are developing algorithm. So what? Suppose if they want to perform, they want specifically pipeline to analyze only cancer data. So they create their own program, writing their own program in any language, either R, C, Java, Perl, and they create algorithm to analyze a particular data of interest. Third one is the data visualization. You produce the data, you just created a, but you cannot show data in the format of alphabets or numbers. You need to visualize is through various graphs. So for this, they are also producing various softwares and tools. Or rather than this, they have another option. They are freely available. There are lots of freely available tools and software to visualize those data and database management. So, in any industry, there are various kind of data, either related to sequencing, related to analysis, related to wet lab work. So, these things are managed by a bioinformatician and data analyst. Fifth one is the method development. So, these tasks are the common. These four tasks are the common ones. So, method development is the master task. For this, you need to improve your programmings. You need to understand the basic scientific statistical concept of every tools and software, so that you can design your own algorithm-based tools and software. So, this is also the main part of bioinformatician and data analysis. So, this industry demands more of this actually. So these are the another aspects of career development in the field of bioinformatics. Research scientist and project manager. So same way, they are just planning their project and management. Means they are designing their own experiments, sample preparation, data integration, analysis and interpretation, and finishing on decide their timelines, budgets, and quality standards. After that, means they will just data analysis and interpretation, Actually, you analyze the data, but the master conclusion is done by your head or your superior one. So that is done by a scientist or any project manager. Next is the publication and grant writing. So you analyze data, you come to the conclusion, but no one knows what you have done. So for this, you need that research scientists and project managers used to start writing papers also on grant writing. So everybody publicly knows that these works are done by your group or done by yourself only. Fourth one is a team management collaboration. This is a major task done by research scientists and project managers. So these are the things, these are the roles you have to play whenever you will get this kind of position. Next slide. Another one is the sales marketing role in the field of NGS companies. So what you have to do, you have to just check the product, you have to come to the market, and based on their interest, you have to convince and achieve the target and sell those products. So first is the customer engagement. Based on the requirement, you have to just share the buffer solution and these technologies, their softwares or tools. The product promotion means you have to organize various seminars, workshop to entrepreneurship and startups in NGS. Actually, right now there are lots of online platforms available which are designing tools and software to analyze the NGS data. So first, technology development. There are lots of IT companies now involved in the field of bioinformatics. They are used to take the bioinformatician because they don't know the concept of biology. So this development part is uh, done by the bioinformatician only and service provision. Actually, entrepreneurs means any entrepreneurs can provide the NGS service companies offering sequencing data analysis. Well, if you understood if you understand the term of NGS data analysis, so you can be that entrepreneur, so that you can analyze their data and you will get the money from them also. Third one is a diagnostic solution. Means based on means there are lots of laboratories. They are starting their NGS research facility. So for this, they want any bioinformatician. They can analyze the NGS data. Example: RML pathology, then Newburgh Supratech. These labs, what they are doing, they are generating data, but they don't know how to analyze them in silico. So for this kind of solution is done by any entrepreneur or any startups. So you can be the one also. So actually, academic and teaching position, you can also get any position in the field of research and grant funding. Means if you have any good research, you can just write a project, you will get the funding. Second is the teaching and mentoring. You can take lectures also, and the third one is the collaborative research because no one is interested. Means no one have that kind of knowledge about bioinformatics. So you, if you learn this NGS data analysis, so you can be a collaborative researcher, or you can get multiple papers. You can get multiple recommendations also, so that you can go anywhere else or any good position. So this can be a good opportunity for you all also. So actually, NGS technician generally in Indian currency we can say they can get less salary. Means they will start from I guess so they will start from less salary, 25. Initially, if you are 25 to 30, based on the location, the salaries are vary. Bioinformatician data analysis, they will get 35 to 30. Means if you are working in Reliance, Accenture, and Bionome, X-rays, so they will provide this kind of salary. Or any research, like if you are working for JRF, SRF, RA, they will provide you good salary. The scientists and project manager have lots of pressure, so they are getting lots of salary, and they are the superior one. Fourth one is the sales and marketing. Actually, sales and marketing is the master one in the field of technology, where you have to just sell the product, and if you achieve the target, they will provide you extra pay also. Factors influencing the NGS data and salary and levels. Actually, education and experience, both the things are different. If you have experience but you don't have education, so that this is the main reason. Suppose education doesn't mean you have to take degrees also. If you have certification, then you will go to the position of research scientist or project manager. So these are the first work in Delhi, Pune, Bangalore or Hyderabad, you will get more salary compared to other cities because there is lots of development in the field of bioinformatics. Other companies are also working, but the pay scale is high over there, or in abroad also. So we can compare any field, but NGS is right now involved in the field of molecular biology, genetics, biotechnology, or any life sciences field. So any researcher want to cooperate with you. So their research shows more importance. So actually, it is very important factor right now. Uh, so if you learn this NGS data analysis, you can earn money at home. You can do work from home jobs. You can you can also work as a freelancer. You can earn. You can manage from home any data analysis services. So this is the best part. You don't need to invest anywhere. Just learn the analysis. Just plan and just give the result. You will get the money easily. Actually, there are various courses like BSc, MSc, and PhD in the field of bioinformatics, but it took lots of years from college and university to get a degree. But here is the shortcut to get this kind of training through this platform. This platform is the BioHealth Learning. Actually, this BioHealth Learning program is the most important program for you all because it runs various programs related to NGS, CRISPR, molecular biology, network biology, drug designing, genetic engineering, what they are doing actually this program is not specific to particular students. It is very useful for students, professionals, and enthusiasts. So if you are non-technical person, you don't know the meaning of programming, you don't understand what is software or database, but here your basic concept must be clear and you will enhance your knowledge, you will improve your skills using the hands-on session. So here this platform provide you theoretical as well as hands-on session. They will provide you workshop also. They will guide you how you can get job. They will guide you where is the vacancy available. So this is a very helpful platform. Provide lectures, hands-on training, as I already told you, interactive sessions, workshops, and future aspects to guide students through the learning processes. And during these various programs, they will also available 24/7 to clear your doubts also. So you can just mail over there. A particular faculty will just guide you which problem you have, and even they will help you in your research program or you just complete your dissertation. So these are the key features of biohealth learning. Comprehensive coverage means it takes fundamental concepts from basic to advanced. Second is the hands-on learning. Actually, this is a major thing which no one will going to explain you. Practical session is also carried out here. They will teach you the basics and they will give you the example how it works. So it is easy to understand and it is easy to design your own work plan. Third one is the interactive approach. Actually, during the various examples, case studies, they will also guide you to for question answers for deeper understanding of subject also. Fourth one is the updated curriculum. Actually, this biohelp learning will help you to improve your CV using this kind of certificates, so that you can just shine in the industry easily. By means you are just NGS data analyst, you can work as an NGS data analyst also. Fifth one is a supportive learning environment. Here they are provide you the guidance, support throughout the course, facilitating a positive learning experience for all the participants. So here are a lot other things carried out by hard learning, which is skill development. So first is the fundamental of biology. Since they are clearing your concepts, biological concept, and the crucial for bioinformatics. They will explain you genetics, molecular biology, genomics, proteomics, and evolutionary biology also. They are also learning another important program. This is programming and data analysis. Here there are another courses related to Python, R, Perl, which are essential for NGS data analysis also. So participants learn how to manipulate biological data, perform statistical analysis, and develop algorithms and scripts for data processing and visualization. Third one is the bioinformatics tools and software, data with a C. Everyone knows the terms of bioinformatics tools and database, but they don't know which one they have to use, one where they have to use, how they have to use. So here we are to help you how you have to use this kind of databases for your work. So NCBI databases, Ensemble, Unifloat, Blast, and others. So this can be how useful for you all during the NGS data analysis. Fourth one is a sequence analysis. So they will provide you the knowledge how to analyze DNA or RNA sequences, and which is useful for motif discovery, phylogenetic analysis, and functional annotation. Structural bioinformatics, they will also provide you the guidance in the field of molecular docking, provide you means molecular docking, protein-ligand interaction using various tools like PyMol, Modeler, Autodoc, NGS data analysis, which already today I given a lecture, which is very trendy and very useful for you all to get in the industry. Seventh one is a data visualization. For this, you are using actually they can learn very informative visual representation using tools like ggplot, matplotlib, and d3.js. For eighth one is the machine learning and data mining. So here you can also learn machine learning platforms, data mining, how participants to develop the predictive models based on machine learning programs and languages, algorithms, classification algorithms, and feature selection methods. So these are the skill development programs also carried by the biohealth learning. So you can also enroll with for that. So it is very useful for your curriculum. Ethical and legal consideration also. The practical projects, hands-on sessions are also carried out using biohealth learning. So during this 15 days NGS data analysis program, I'm going to tell you the overall broad analysis project. Means we'll start from the introduction to NGS technology, where you can just learn the features, their application, their workflows. Then in second thing, we will just go through the library preparation of NGS, means how the library works. Means you can just selecting the and samples. After that, you have just isolating them. Then proceed for the library preparation. Third one is the NGS data generation, how that instrument works. Now, fourth one is the very important hands-on session starting. So here you can just learn various file formats of NGS data analysis, how we can proceed for NGS data pre-processing and quality control using R language, using online available various softwares and tools, which I will explain in this course. Fifth one is the NGS data analysis part 2, where you will going to learn variant calling and identification, annotation, functional analysis of variant, introduction to variant calling algorithms, and hands-on session using case study for variant calling and annotation using R language and another softwares and tools. Sixth one is the transcriptome analysis, where you can just retrieve the RNA sequence data, perform the differential gene expression analysis, pathway analysis, and functional interpretation. See, here is the NGS. But inside this, we are providing you hands-on session so that you can your NGS data analysis concept is clear. NGS is not specific. It is related to variant analysis. It is related to transcriptome analysis. And further, I am going to explain. 7 day session is the epigenomics analysis, where you can just understood the epigenomics data identification, which is useful for the identification of DNA methylation. There is a hands-on session on that. Then metagenomic analysis, which is useful for the environmental biodiversity understanding. We will also have an hands-on session on metagenomic analysis. Ninth one is the cancer genomics, where we are going to work on a cancer data. We can analyze the cancer data sets and we'll come to the conclusion. Tenth one is the structural variant analysis. So here the structural variants. You can just understood various algorithm structural variant analysis carried out through the algorithm tools. So you can understood the particular changes in normal one or disease one. And single cell genomics in on day 11, and day 12 we will just visual. After this analysis we are going to visualize. So the after from day 5th to day 12 and day 13, we have hands-on session. It's about 7 to 8 days related to NGS analysis, single cell genomics, transcriptomic, etc. So on day 14, I will just brief about the NGS data analysis steps, guidelines, doubts, any concern, any question, any query. And 15, there is a closing session for this course, and this you will just clear. I hope you all have this clearing thought, clear concept of NGS data analysis, what you have to want to work, which work you want to do in the field of NGS data analysis. So this is all about the 15 days program uh, running by BioHelp Learning. It is actually very important for you all because now everyone wants to involve in the field of NGS because it is a basic and must requirement. If you don't have a degree, it is not required right now. If you have a certification, so you can enroll anywhere. You can work in industry, in research, at academic and lab, anywhere. But you have some clear concept about that. So, actually, this certification does not require that much cost because if you are going to take degree, it will take lots of money. But the certification is just carrying 1,200 rupees course. It is 15 days program. We will provide you hands-on session, the theoretical example. So I think if some of you are researcher or some of you are graduate or masters, so they can design their own dissertation or project easily. So we will provide you some internship opportunity for particular places also, which are available in under our sources, and we will provide you some supplementary materials, tutorials from where you can further learn or further develop another your prospects based on NGS data analysis. So this is all about it. And this 15 days program is actually eye-opening session for you all and understood the basics to advanced concept of NGS data analysis. Thank you all. If you all have any doubt or any query, you can just message me. Hello. So I will again very thankful to you all to attending this webinar, and I am actually very I am also not new to this field. Actually, I'm working since 8 years in this field. From Miss, we have already we already design our own work plan of PhD. So if you are going to join this program, so definitely I am going to help you in any way of your NGS data analysis work. Even it is very helpful to join in any industry. We'll guide you. We'll provide you guidance also. So, thank you, all of you. Thank you once again for this webinar. I hope to meet you all during this course. Thank you. Thank you, everyone. And for this program, you can just directly go to the website, just contact to the sir. He will guide you from which day it is going to start. So, just ask them. And this 15 days program is really very helpful for you all to understand and clear the concept of NGS data analysis. And now, thank you everyone.